Okay, well, we'll get started. Welcome everybody to the first post-Thanksgiving IMAG MSM Working Group mini-seminar on virus modeling and immune modeling. We have two great speakers lined up for today, Jacob Barhak and Gary Ann. As usual, I need to tell you that the meeting will be streamed and recorded and will be made available. So please don't say anything you don't want to have in the archives. We'll have a bit of a discussion afterwards about the organization of the working group. So I hope people can stay to have that discussion. Just remind you that Reinhard Labenbacher and I are the co-leads of the group. Jim Sluka is their web administrator, and you've probably had emails from Bruce Shapiro, and Reinhard and I are people to talk to on anything related to content of the working group. Jim about the website and anything there, and Bruce on any organizational issues. We have a Slack channel. Please feel free to use it. We probably are all saturated, but please help us maintain communications. So without any more delay, we'll start. Our first speaker for about 15 minutes is Jacob Barhak. He's going to talk about reference model accumulates knowledge with human interpretation. Jacob, are you ready to queue up? I see you there.
Yeah, I will. Yeah, okay. I will also add some information for everyone. This is how you can get it. So I will add some more data over there little by little because it takes a while. You can find in the chat some references for later on that I'm going to talk about. But for now, oops. Yeah, and I'm just want you to have those available. So you can browse this information, and some of it is already related to IMAG. So I wanted you to have all those links. Let me share my screen. And what you're seeing here is the presentation. It's the first link I sent you. It was supposed to be a poster for the last IMAG meeting, which was delayed by the pandemic. It was supposed to be about the diabetes model and the new inventions there, but now it's actually applicable for COVID, and I'm going to show it near the end. I'm talking about the reference model for disease progression, and the reference model started because I saw the models for diabetes, and I saw that you need to be able to combine multiple of those together, some sort of an ensemble model. So in 2012, I started developing it. What you see here is the diagram of the diabetes model. It's a state transition model, but notice the states are in different processes and the processes compete. Let me explain. If a person dies out of a heart disease, MI, they cannot die out of a stroke afterwards, and vice versa. Also, if they get hit by a bus, which is a competing death, or by another disease, then they cannot die from those conditions, and other conditions cannot develop. So this is a race that's coming here, and the model is able to do this and compute it because it runs micro simulation, very similar to agent-based simulation. And it runs for each person. It stochastically runs what might happen to it, to that person using Monte Carlo techniques. However, you don't do this for one person. You do this for many people and for multiple populations of multiple populations. Each one has many people inside it. And the information is heterogeneous because people are different. And when you run this for a model, you need a lot of computing power if you do it with many people. But I actually need more computing power because I'm not running it for one model. Each one of the transitions you see here in this diagram can be governed by a different equation, and this equation can depend on other states that are in other processes. So there's a mix of things that are happening, and this gets governed by multiple equations that kind of interact with each other, and they all together become an ensemble. I started as these equations competing against each other, but later on it became an ensemble of those. Now, having an ensemble is one thing, but what do you do with it? Well, the reference model is called a reference model because it has references that you have to adhere to. First of all, the models, where they come from, and the models are kind of assumptions. But then from the other perspective, you have something that you need to compare the simulation results for. So I take clinical trials that were done for diabetes, and I compare the model that was executed on a specific clinical trial population and compare it to the end result of the clinical trial. And I create a reference where I can actually later on show you how well or how unwell each model behaved. This technology is patented. So unlike many of you, I'm not academic. I left academia to pursue this, and in the last 8 years developed this technology. This technology is now patented. So if you're doing or need to do something similar, talk to me. I also have a conflict of interest because I'm not an academic and I want to declare it. It's important for you to know. I contract with other companies to do the work. So therefore, whatever I'm showing you may be influenced by other things, but I try to be as just as I can. You'll find all sorts of information about what I do in my website, and as transparent as I can be. And also you'll find in my publications a lot longer, long conflict of interest declaration. Go back to the reference model itself. In 2012, I started it. Each year I added pieces of technology that enhanced it a little today. So in 2012, it started. 2013, I added cloud computing and high performance computing, a lot of computing power to run all those models against all those populations, especially in micro simulation level. I added evolutionary computation to better mimic the populations I'm modeling. And then object-oriented, because I had so many populations, you had to organize it. I added the assumption engine. Basically, this is the engine that drives the ensemble model. In 2016, what you're seeing here is the number of models or model combinations that I was running. So you can see at this point, it's infinite number of models. Basically, if you have a set of models and you have a modeling space, it kind of becomes infinite and you need an ensemble to resolve it. Because although the basis functions there are only a few of those, when you combine them, they become infinite number of combinations in an ensemble. 2017, I connected to clinicaltrials.gov. This is why you see the jump here. This is the number of validation points I'm validating against. So 2000, it get to about 360. I started from about 150. This is where I connected to clinicaltrials.gov. You see the jump, but also I continued this, added some graphics, and this year I added human interpretation, which I'm going to talk about in a second. Today I'm at about almost 500 validations. This is the number of populations I was modeling, and you can see this is number of populations, the cohorts, and again, the validations. And you can see how this number grew throughout the years. Now, after I have an interface to clinicaltrials.gov, it's much, much easier. Now, let's talk about the equations. Notice there are many types of equations that on anything that can affect diabetes, how biomarkers change, how there are multiple death equations, and multiple types of death equations as well. MI equations, stroke equations, the number of those, I stacked up all the equations and put them together. This is the 2012 model. And you can see how the number has risen throughout the years. This addition of interpretations, I added 6 interpretations of so-called experts. Those are not real people. Those are, think about it, a person who would think like this is considered an expert. And I added 6 of those. And I want to focus exactly what I mean here, because this is the new part that I was talking about. I want to explain. So when you conduct a clinical trial, you eventually have reached some endpoints, and those endpoints are either single endpoints or composite endpoints. Let's say if a person has an MI or heart attack, it's usually a single endpoint. If they reached it, then they count it as one if they reached that condition. However, you can combine them. So for example, cardiovascular disease is combined from MI, stroke, and some other conditions. those definitions, even within trials that are conducted in different times or different places, or even the same environments, are sometimes different. So the numbers reported in the clinical trial represent slightly different measurements. Ideally, those will be reported as ICD codes. However, not all trial reporters and ICD codes look at clinical trial publications. It's becoming better, but it's not there yet. And even if there are ICD codes, the ICD codes not always match between the definitions of the trials. And of course, there are multiple levels of multiple versions of ICD codes, and you need conversions, and things get lost in conversion. So eventually, you need a human to look at those numbers and tell you what those numbers mean. Are they overestimated compared to what your model is calculating, underestimating? Also, one more thing, some definitions change. I gave sepsis as an example, but sepsis definition has changed throughout the years. Gary Ann is after me. I saw a lot of paper on sepsis that he wrote, so he will confirm it. Sepsis has changed throughout the years, the definition of sepsis. Same thing happens with other things. So if you have older clinical trials and newer clinical trials, you need some level of interpretation, and some study actually are aware of it. So when they publish the study at the end, and I'm giving the example of the record study, they actually tell you if we count our endpoints like this, or we count them in a different way, then the numbers we get are slightly different. And they publish this. This is part of the publications. They are honest about that, which is good. So if my model counts an MI according to one definition, and especially if an ensemble of models, they can come from different definitions. When you combine them together, you eventually need something to, some filter to, before you do the comparison to the observed data from the clinical trials, you need some buffer, and this is human interpretation. And I'm adding it this year, just like the models, you have multiple human interpretations. Think about the situation in COVID time. Even now, they report numbers of infections per state or numbers of deaths per state. Are those numbers the same per each state? So first of all, there are errors that they find every once in a while and correct. There's all sorts of anecdotes about that. But beyond that, how did they reach the infection number? Did they is it a percentage of the number of people they tested? And did they test everyone in a proper way, or is it only the number of positives that they found in their test, which maybe the test sample is smaller than the population? All of those questions are relevant, and you need an expert to come in the middle and says, I believe those numbers, or I think those numbers are overestimated or underestimated. Same thing with death. If someone had COVID the minute they die, did they die out of this disease, or maybe they just died of something else, but they said that they had the virus and therefore they were registered as COVID. So those are questions that an expert can give you some interpretation. Let me show you how this works. The reference model actually takes all of those equations that I discussed before and gives them a weight at the beginning. At the beginning, you believe every equation that you gave and each interpretation of each expert the same way. This is why you see those bars with a certain color, the same height. This is the influence of each model. You can see the MI models, stroke models, also mortality models, and human interpretations over here in cyan. What you see here in the upper left graph is the clinical trials. This is like acronyms of represent the clinical trials and the cohorts. So you can see the cohorts over here. There are statistics about what's the average blood pressure, how many were smoking, and so on and so forth. You can play with the display a little bit, but this is less interesting for now. It's more important to explain what the heights are. What you see, the height of the circle, it is the error between the model prediction, the ensemble model, and the actual observed results of the clinical trial after interpretation. So here you're seeing the combined interpretation, but let's look at expert number one. Expert number one in my situation here says, okay, I believe all the numbers reported are defined exactly like the model. You don't need to add any buffer in the middle. No human interpretation is needed. This is what the model therefore would see. The model will try to optimize, and what you see here is the convergent curve. We'll try to optimize this, and you can see all of those, each iteration, and each one of those iterations takes a long time, even on high performance computing machines. Each iteration, you'll see some sort of fluctuation and improvement, gradual improvement towards convergence. And in the bottom, you see how all of those bars change height, and you can see some of the bars disappear. Those are models that will not be used by the ensemble. The rest of the models some of them become dominant and say, okay, this model represents well with all other models and other interpretations represent the best what's going on in this situation, the question that you ask the machine, the query that you ask the machine. However, notice that different experts will see the same results differently, and this is something that we can observe and look at around when we change the expert. However, the combined experts is the one that takes those experts in those weights, believes them this way. So for example, the most believable expert is expert 6. He's about 50% of his prediction is used, about 40% from expert number 4, and about 15% from the last one, which is number 2. The last 3, the system tells you probably ignore them because the information they provided doesn't match well. So the same thing that I applied to diabetes can now be applied to COVID. This is a recent publication a few months ago about the initial use case. You can find it in Curas. I sent you the link. This is the diabetes model. This is how the initial model for COVID, this was how it was published there. For COVID, I'm doing something very similar, but I'm doing it for US states, not for clinical trials. And I combine the infections and deaths, and the fitness score here shows the difference between infections and death per state. When I run the model, and here you see the convergence on the bottom, you can see the convergence plot, and on top you've seen some of those outliers drop down to the ground. This means the model got optimized, and what I'm optimizing is parameters. The model here is very, 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 very simple, very similar to an SIR model with a touch of ensemble just for the mortality. I have 2 mortality models, both derived from the CDC, high and low, because this was my initial use case. Now I'm working on a more advanced model. Maybe it's worthwhile saying that the model results that you see in here, those are computed, just to give you an idea, 9 hours on the Rescale cloud. Rescale gave their infrastructure because of the pandemic. They donated their resources. Microsoft Azure gave money, so I was able to run it. It takes 9 hours for 880 cores on 20 machines on the Rescale cloud to compute that. So it's a lot of computations. This is now a model that John Rice is here. This model is currently running on a server that he hosts today. And also I want to thank Jared Hodges for taking care of the simulation there. And the model here is the current COVID model. These are not final results. This is just to show you the flavor, because I want to ask you for help from the working groups. I actually do need help from some of you for some things. What you're seeing here is the same idea as before. We have multiple models though, however. The reds represent infection models. For example, infectiousness curve. This is why I asked Alan Perelson for a few weeks ago. He gave a talk here in this working group, and I started communicating with him. Maybe I will able to plug as an infectiousness curve in one of those infectious models. If you have more of those infectious models, please do contact me. I'm interested in plugging them in. There are transmission models here. How do the models transmit? What you see here are response models, how people respond to the pandemic, how they behave. Those are behavioral models, the cyan, and the rest are mortality models. But more importantly, I currently don't have experts that will interpret the numbers comes from the US data. I'm showing here every 5 days the numbers that come from the COVID tracking project. And my last publication, this publication, both take data from the COVID tracking project, so I want to thank them as well. At this point, I am looking for your help I will stop the talking and I will ask you for questions, and hopefully you have questions that say I can help.
Great, thank you very much, Jacob. I think that your focus on aggregating information from different sources and connecting to clinically meaningful results is important. We have time. We have to keep moving a little bit so Gary can speak, and we have a little time for discussion. But do we have time for one question or two questions in the room?
So let me just ask Jacob, because you sent an email to us. So what exactly do you want? What sort of community engagement in terms of it's because it sounds like you have data. So do you need these kinds of population level or anything? I'm just trying to figure out what it is that you need input on.
So population level, the population comes, the nice thing about a reference model doesn't need the individual data. It can live out of data that is per population statistics. So I have the ability to merge those statistics. So I'm not looking for the population statistics unless we want to add more populations for different countries, which is a possibility. Some people talk to me about this, but from a research perspective, it's interesting to compare different countries or different behaviors, but I'm more focused currently within this working group on things that people actually develop. So for example, when Alan Perelson talked about, I think it was 3 weeks ago or a month ago, he gave a talk about how he has the he has the infectiousness curve within the upper respiratory system and lower respiratory system, and how much virus is there, the viral load. When he talked about those, I sent him an email and he tells me, oh, here's a paper, and he actually has an infectiousness curve equation there. Says, oh, the person is infectious after transmission this much. And he has several things there. So I'm trying to incorporate this equation. I'll have to talk to him more, but this is one example of things I'm looking at now. And I know some people already send me something similar. I need some of those that I can actually plug in, and then those assumptions go into the model, and the model will compose those together and tell you which one fits better the data that you have. And the data can come multiple sources. The other part is the human interpretation. If someone looks at all of those per state numbers like deaths and infections and can tell you this is overestimated, underestimated, this is data that I can use to plug into the model as an interpretation. It's a human interpretation thing. It doesn't have to be correct. It has to be on human intuition, or maybe sometimes it's human, it's what they just human thinks, and sometimes it's right, sometimes it's not so right. But the more of these I have, the better. And if you have other types of models, we can talk. Maybe they can fit as well. The ensemble model just accumulates information from multiple sources. Did I answer?
Yes, thanks, Jacob.
Okay, thank you, Jacob. I think we better move on. I hope people can talk offline, have fruitful discussion. Thank you again, Jacob, for being willing to speak. Now we'll move on to Gary Ann's talk. That's a lot of topics to cover in a short talk. So
yeah, so it's actually going to be mostly on the first part. So I can't start sharing the screen while someone else is sharing. There we go. Okay. Try. Okay, so the title is a mouthful. Can you guys see? Let me just, it's black right now, right?
No, we're seeing your presenter view, so you probably want to reshare the presentation.
Is okay, so it's going into presentation mode. There, is that it?
We're seeing it in presenter view. You need to reshare to show the audience view.
Let's see.
I mean, it's fine, but we see your desktop essentially, not the presentation.
Okay. How's that? That better?
There you go.
Okay. All right. Yeah, so a mouthful of the title. Just for those of you who don't know me, I'm actually a clinician. I'm a surgeon, critical care physician. So I've been doing a lot of this computational stuff, but a lot of it is focused on trying to improve kind of what we're doing in the clinic. May not necessarily be directly related, but in general, I have this very translational bent. So that kind of is a bit of a framing for what I'm wanting to talk about. So, obviously we're dealing with COVID right now, and, I'm probably not alone in having some frustration about the relative limited number of things that we could do to try and mitigate, the response to the pandemic. Basically, we're stuck with public health non-pharmaceutical interventions, which, of course, are nice, but there's a huge challenge with respect to what to actually do in terms of people who get sick. The management of the sickest patients has significantly improved as people learned how to take care of COVID-related respiratory failure. The steroids is definitely, I think, a positive. And then there's an entire alphabet soup of cytokine type mediators that are out that is one of the things I want to talk about. So, this challenge number one is the limitations of what we can do. The first one I mentioned about. The second one is coming up with novel antivirals is really, really hard, because coming up with antivirals is really, really hard. And vaccines take time. And, the companies that are rolling out the vaccines now have done, incredible work from that standpoint, but it is essentially a time-consuming process. So the challenge in my mind is, what can we do between, the public health, non-pharmaceutical interventions and vaccines? And we saw a lot of repurposing of existing drugs. How would they work? It was essentially done empirically. In many circumstances, a very haphazard way that led to a lot of confusion and, I think, misinformation with respect to what could be done. I think in preparing for how to actually treat some of these patients, gain some sort of insight into the pathophysiology of candidate pandemic viruses, right, which is a comparative virology zoonotic reservoir challenge. And then shorten this time to effective vaccines, which is accelerating the pipeline. We'll talk about that. Right, so my proposal, and hopefully is shared within this working group, is that the responses to challenge number two can be accomplished by building a repository and infrastructure for multi-scale modeling to aid in these particular processes, these particular steps. Right, so just a little bit of my modeling credibility here. These are my primary focus has been, as Jacob mentioned, in bacterial sepsis, systemic inflammation. This innate immune response, ABM, is a model that's about 15 years old, 20 years old now. We have multi tissue models. So that was just
sorry, your screen black, Gary.
Okay, I'll stop that one. And then we repurposed a existing model of that one died too. Right, so it doesn't like me running movies. That's okay.
At least me, I'm only seeing the whole screen black.
Okay,
oh.
Hang on a second. It's these movies have occupied the bandwidth. Let me go back.
Yeah, it's back.
Okay. And then I'm going to focus talking about this innate immune response agent-based model, which we've done in silico trials of multi-mediator therapy of the type that have been tried with respect to the COVID cytokine storm. And we'll jump off that in a second. Our current model that we're working on is the cellular immunity agent-based model, which is essentially the innate immune response model with the cellular immune components added into it. Here's a wireframe diagram of that particular model, shows some of the various components associated. It's obviously pretty complicated. I think there's been some discussion previously about, you know, simpler versus more complicated models, and the challenge of parameterizing these types of things, which is exactly the problem. And that's what's going to form the bulk of this talk. I would contend that that if you're going to try and come up with interventions or want to evaluate existing interventions, you need a model of the same level of resolution so that you are limiting the number of inferences you're making about the effect of that particular intervention. And these particular circulated mediators with the CIABM represent cytokines for which there exist, pharmaceutical grade monoclonal antibodies or antagonists, and probably were tried during the COVID epidemic. So I would contend that in order to evaluate somehow prospectively what combination or when these types of interventions should be applied, having a model of the level of resolution that incorporates the quasi-mechanistic effects of these components is necessary. Right, so this gets to the issue of biological heterogeneity, and this is something that all of us deal with. It's a difference between species, it's a difference between individuals, and importantly, with respect to how we calibrate and ostensibly validate our models, the difference between samples, right, as manifested in the inevitable error bars associated with any sort of data collection. And traditionally, biology is focused on the differences between particular systems, right, with ever, microscopic subspecialization of things. But, this is kind of a recurrent theme, and what I'm interested in is that there must be some supposition of similarity from one system to another that allows you to take knowledge extracted at one level and apply it to another context, right? And I've written about, how the role of multi-scale modeling in crossing this particular translational divide in this little opinion piece in the Bulletin of Mathematical Biology. With this idea of using multi-scale models as conserved pieces of knowledge that allow you to contextualize data from one circumstance to another is a crucial point. And so when I showed you that diagram, the CIABM, each one of those arrows represents a rule. That rule incorporates a whole series of parameters, and we know that there's a huge challenge in the parameterization of these models. And in many circumstances, the goal of many modelers is to try and lock down these parameters as much as possible. With, I think, the negative side effect of creating very brittle, non-scalable, non-generalizable models that miss the richness of the heterogeneity that's present in the biological data, right? Because assuming we don't have measurement error, the thing that we know about a series of data across a set of error bars is that the system was able to generate that data. Something about that system was able to produce that. So when I think about at least how we convert biological knowledge into computational rules for a model like an agent-based model, it follows along this kind of very simplistic generic pathway representation above. And while we would write this this relationship in a certain sort of way, with an arrow going from one component to another component, the fact of the matter is that there's a whole host of cellular machinery that affects that particular state transition that's not explicitly represented in that particular pathway representation. That could be genetic variation that affects the effectiveness of some of the signaling enzymes or the processing components, or hidden pathways that exercise feedback control that haven't necessarily been characterized. So thinking about it in that sort of way allows us to write that particular pathway with these particular parameters, where the parameters themselves represent this host of uncertainty that we know exists, but we cannot quantify within the context of that particular representation. So if we take all those parameters and all those rules and create a matrix out of that, we essentially have a matrix of the coefficients of all the rules within all the components of that model. And that is something that we call the model rule matrix. I want to just say, I want to thank Chase Cockrell, who's a researcher I work with coming up with this idea and doing a lot of the work that I'm showing here. Right, so we assert that this model rule matrix and the terms within that represents essentially the sum total of the genetic epigenetic variability unknown regulators on the particular rule structure that we are representing in that particular model. And therefore, this rule form can represent, the rule form can represent what's conserved between one circumstance to another circumstance, be it between individuals or actually species. And it takes into it makes explicit this assumption of similarity that we make whenever we interpret results from one experimental model to another experimental model. So like if we study neutrophil activation in a mouse, we assume that there's something similar with respect to how that occurs in the human being. Right, and my contention is if we want to try and get clinically relevant populations, you need to operate over the entire plausible space of these parameterizations, which is an exponential computational challenge. So the first thing is that how do we refine what this model rule matrix is based on data? And then this goes back to the idea that if you look at a set of experimental data that contains error bars, that's heterogeneity within that, and therefore even the outliers within those particular error bars, again, assuming you don't have measurement error, are somehow reachable by that system that is being sampled. Right, so in many circumstances, when you try and fit parameters, you kind of throw away the outliers, you draw a regression mean through the center of the data, and that washes out a lot of information about how that system can actually function. This is important when you're trying to deal with clinical populations that manifest all that heterogeneity. So what we've done instead is looked at a way to operate across the parameter space represented by the model rule matrix as a means of encompassing data that is actually present, right? So if we go back to the idea that the model rule matrix, the MRM, is a representation of all the genetic, epigenetic, and unknown variability associated with a particular pathway, right? If we operate across that entire thing to find out which combinations can actually produce data that's in the real world, that entire bioplausible space is something that we would say, if we want to design a robust control for that system, we need to operate over that particular system, right? And that avoids trying to focus too much on a particular representation of the model and trying to be more generic. So what we see here in the left lower corner is how the optimization process of fitting the heterogeneous data enriches what the rule matrix looks like. So the rule matrix on the left is inherently sparse because when we write what those rules are, there's only a finite number of interactions that we define. But this demonstrates an example of applying this model rule matrix enhancement to heterogeneous data. What you see on the right here, the range of data generated from a clinical burn population in terms of cytokine time series, and fitting to the heterogeneity of that data, right, or encompassing the heterogeneity of that data provides a much richer optimized rule matrix, which actually looks much more similar to a gene expression profile, right? So that is a kind of the idea that what you're doing is you are enriching what the interactions are within your model by doing this process. Again, the idea is that you're not fitting to a subset, but you're encompassing the range. So as opposed to overfitting, this is actually a robustness aimed process. So once we get this entire huge space, then we need to figure out what are we going to do within that particular space. So the parameter space that we have for when we run our simulations not only includes the coefficients of the rules instantiated in the model rule matrix, but a whole series of external variables that are associated with the perturbation that we're applying. So what is the viral load? What is the virulence of a particular infectious agent? What is the transmission, likelihood, or what is the life cycle of that particular thing? Right, so when we think about a simulation and the parameters associated with that particular simulation, there are the internal parameters, which are essentially the coefficients for the rules, and then the external parameters, which are property of the perturbations. Now, if we're thinking about trying to characterize a clinically relevant region of parameter space, we need to operate over both of these parameter spaces. And that's a extremely computationally intensive, intractable computational task if you try to do it from a brute force standpoint. So we have adopted this nested active learning method, which is a machine learning method that provides a gradient-directed search for appropriate characteristics of global parameter space that you're looking for. And this is something that we do to identify the subset of parameters that we will consider to be representative of clinical population that we will then do control discovery on top of using either genetic algorithms or deep reinforcement learning. Right, so a quick sidebar into deep reinforcement learning. Most people know about this through the DeepMind projects, AlphaGo, AlphaStar, et cetera. These are model-based DRLs where there is a simulation model that is the basis for the deep reinforcement learning task. We applied this to the IRABM, first circumstance in biomedicine that I'm aware of model-based DRL. Here are some characteristics of how we set up the training process. Without going into too many details, the fact is that we could take a set of conditions with a 46% mortality and come up with an intervention policy manipulating the cytokines within that particular system to make that mortality 0, right? And one of the keys here is this initial project involved simulations with antibiotics because we were simulating bacterial sepsis for which antibiotics are part of the treatment. As I mentioned before, that's not the circumstance with COVID, where we do not actually have an effective anti-infective agent, and therefore we reran our DRL with this particular condition. So again, the limitation here is the IRABM is not set up to specifically simulate viral infections, but this is what it would be if you relied purely on the immune system to recover from an infection. So the upper graphs in each one of these panels represents what's happening in terms of the actions that are taken by the AI agent trained from the DRL. And the lower one are with antibiotics. So you can see that there are considerably different policies that the AI agent comes up with, which is not surprising. It also turns out to be a more difficult problem. We take a 75 to 85% mortality cohort and turn it down to 25% mortality, which is not terrible, but it's not the best. And it also shows that the policy controls are more complicated in terms of the non-antibiotic circumstance, represented, like, for instance, the difference between the interferon gamma with the no antibiotics versus with antibiotics. Another thing that's very interesting about this is if you look at the two lower, larger panels, one is effect on interleukin number 1, of which the with antibiotics, did a fair amount of activity with. Versus the action on soluble IL-1 receptor, which is an intrinsic antagonist to IL-1. And you come up with very divergent amounts of focus on either one of these two components, the same pathway. So we believe that this is an example of underspecification that's intrinsic to ML and DRL, and that was the subject of a big preprint coming from Google a couple of weeks ago. Because part of the problem when we do these trainings of these AIs or doing machine learning is we have no idea as to whether or not the convergence is based on the same factors that are present or not. So the difference between the antibiotic and the no antibiotic, focusing on different components of that particular pathway, it's unclear whether or not that actually is a real difference, or is that some sort of stochastic process that came out of the training. All right, so now I'm just going to fly over the three things that we talked about in my title. The first one is this idea of gaining insight about the potential viral pathogens by studying zoonotic transfer. Right, so we know that these zoonotic reservoirs are the source of these novel viral pandemics, and there are extensive research being done in terms of like ecological habitat disruptions that would lead to the transmission of the jumping of potential pandemic viruses into the human populations. I think one of the challenges is identifying what is it that makes that jump actually happen, right? That's an extensive area of discussion as well. It's not unreasonable, however, to think that somehow there might be some insight gained in the pathophysiology of the human disease by looking at what it is the difference between the reservoir and a human, and seeing how a virus with certain capabilities is not lethal to a particular zoonotic reservoir, but then becomes pathogenic in human beings. And this can potentially help predict the manifestation of pathophysiology that would lead to non-antiviral specific, but more host modulation strategies. Right, so I took these two graphs from this Frontiers review on bat immunology, and looked at these two particular areas where bats have dampened interferon response to RNA viruses, and also a dampened response to extracellular DNA or damage-associated molecular patterns. Both of these things lead bats not to have as intensive an immune inflammatory response to the presence of these viruses. It is the increased host response to these viruses that is the basis of the pathophysiologic manifestations that we see in COVID. Right, so the question is whether or not knowing more about how these viruses exist within their zoonotic reservoirs can provide us potential understanding into how that disease may manifest in a human being. And I think the hemorrhagic fevers also have similar characteristics like Ebola and the Marburg virus. And then finally, we're going to talk briefly about accelerating vaccine development. Fortunately, Moderna and Pfizer and maybe AstraZeneca have done yeoman's work in getting these viruses to potential delivery we have to all accept, at an unprecedented level of efficiency and potential efficacy. Right. However, we can still potentially enhance that particular pathway by looking at how vaccines actually work to begin with, right? To filter candidates and identify systemic and off-target effects, importantly, which are maybe related to some of the vector issues. Right, the CIABM that I mentioned earlier was originally developed to evaluate development of cellular and T-cell immunity in the dengue setting. But it goes without saying, to a certain degree, modeling immunity requires modeling infection. And we have, in a very preliminary fashion, looked at matching the CIABM to influenza data. It's not great data, it's in the preprint form, but this is all work under evaluation. I think part of the message that I would hope to get across to this group is that we all become very enamored with our own particular models. I particularly am not assuming that my model is any better than anyone else's model. I think that having a range of these types of things is important. This goes a little bit to Jacob's ensemble idea. And the hope is that this particular working group can help develop a multi-scale modeling repository or resource that can provide biothreat response capabilities across a range of responses, be it going at the public health level in terms of optimizing and targeting interdiction or public health strategies, down to vaccine development. But I think there's very important middle zone of using repurposed, having some idea about what the pathophysiology and the host is going to be, and having some platform in a more rational fashion to kind of shuffle around all these combinations of things that people are just trying empirically. The other thing I would hope to get across in this working group is the importance of embracing the heterogeneity that's present in the biological systems we're studying and the data that's collected, not trying to necessarily wash that away and focus on things like parameter sensitivity analysis or necessarily prediction across an experimental platform, but understanding that necessary translational step into the human population, the clinical population, involves an explosion of heterogeneity with respect to the response capabilities of the individuals doing that. So with that in mind, one of the subgroups that I'm hoping to help lead, this modeling individual disease and response to treatment subgroup, I wanted to try and emphasize some of the development of methods to deal with this particular heterogeneity, provide methodological support to the other groups as they are doing their particular more focused modeling projects, and use those models as use cases for method testing. So I'm just going to finish by saying that I am a guest editor on this Frontiers in Physiology research topic, which is the integration of machine learning and computer simulation to solve complex physiological and medical questions. Ignore the abstract deadline. Basically, the papers are due in April. I would encourage any of you, and I think the MSM community is very well positioned to do this, to send us your work that applies various machine learning techniques either to analyze or to enrich your particular models. And I can tell you that you would get a favorable review, at least from an editorial standpoint, a empathetic review process. So with that, sorry if I ran a little bit over. I'll take any questions that you might have.
Okay. Time maybe for one question. I'm afraid we're on a tight schedule, but Gary talked about so many important issues that this working group could address. I hope if we can't have all of the discussion today online, at least we can start discussion in subsequent and offline channels. Time for one question before we move on.
I can reciprocate and ask a question, but I like your conclusions. But more importantly, you show a slide with mortality. At this point, you lost me a little bit. Can you go back and explain whose mortality of what? Is the virus mortality, the cell mortality? This is why I was confused.
Yeah, simulated patients. So the modeling that I do is operating at a much more under the skin scale than the types of things that many of the things that you're dealing with are looking at, right? So we arbitrarily set a criteria for mortality within the simulations. So 80% of the total system, if that's damaged, we consider that system to be dead. So that is basically the mortality within the context of the simulation experiments. There's no necessarily correlation to any sort of clinical population.
Okay, got it. Thank you.
Okay, thank you very much, Gary. I hope again that there'll be plenty of opportunities. Use the Slack channel and the website and email, of course, to talk. And thank you again, Gary, for covering so many important topics. Next week, we have Amber Smith speaking. We have an open slot if somebody is interested. And then 2 speakers from NIAID on the 17th. And Filippo Castiglione. Then no talks on the Christmas Eve or New Year's Eve. And then in January, continuing with Paul Macklin and Bard Ermentrout. Grace has agreed to speak to us on January 14th. Again, please suggest speakers. They don't have to be working group members. We'd be very grateful. I'll turn it over now to Reinhard for the last few minutes to update us on the subgroup status, where we are in terms of assembling subgroups, encouraging people to start the subgroups doing their jobs, some of which are happening. Reinhard, over to you.
Great, thank you, James, and thank you, Gary and Jacob. Many thought-provoking ideas, and hopefully, as James says, these can be continued to be discussed both in the subgroups and the working group as a whole. So in the remaining 3 minutes, let me just say a few things about the subgroups. Of course, you know, any work that will get done within this working group ultimately will have to happen within subgroups. So as you might have seen, and here on the slides, you can see there is 30 subgroups that have been formed. So that's, it's great to see that much enthusiasm for different aspects of this. On the other hand, 30 subgroups is a lot. What might be happening is that over the next maybe month or two, some of the subgroups will realize that, you know, maybe they're best integrated with others, or maybe there's not really a good basis to continue at the moment. But in any case, I think it's great that there's so much interest in this. So there's 2 slides. The next one, if you go on, there are 2 subgroups for which at the moment we don't have leads. And there are quite a number of people signed up as members. So hopefully somebody will look at this and take on these orphan subgroups as leaders. I think it'd be great if we could populate those. Also, if you have not signed up as a member or a lead for any one of these, please do so. And the subgroups now need to organize around certain topics. Going forward, if you want to create a new subgroup or combine subgroups, you know, contact us and let us know why you would want to do that, and we can discuss that. So I think this is sort of an emerging structure, these subgroups. So let me just, in the last minute, if you could go on to what these subgroups are actually going to do. If you look at the proposal that James and I put together for the MSM steering group, there are several things to be done, several deliverables that we would like, and these deliverables would come from each of the subgroups. The very initial one is to work out a plan to achieve these objectives, and then you have 2 of them. One is essentially, you know, sort of make a survey of the particular topic that your subgroup focuses on, and the second one, if you go one more slide down, and the second one really is to put together a white paper. It could ultimately be an article, depending on where we want to take this, that essentially says, you know, what's the state of the art, what is needed, and what do we need to do in order to get there, right? So the way I always think of this is any one of these white papers should be snapped up by an NIH program officer saying, yes, we will design an RFA around that, because it's a really coherent and obviously impactful research program. So that's what I think about it. And then, of course, the other function of these subgroups and of the working group as a whole is to catalyze research projects through presentations, discussions, whatever else. You know, maybe post-COVID, one could consider having an in-person workshop in either the whole working group or some subgroups. So there's a number of things that one can do to make these live on in terms of a rationale. But these are the objectives. So what we would like from you is ideally feedback on both the subgroup structure and the objectives, so we can finalize that and all get to work. And so that's it, with -1 minute left. Thank you very much for joining us, and we'll reconvene next week unless there are any short comments now.
Well, I was going to say, since we have 1 speaker next week, we may have a little more time for discussion. If the subgroup leaders could be willing to present or talk for just a minute or two about their plans and what they've been able to do between now and next week, we'll have the opportunity to talk a little bit about the subgroups in more detail. I think it might be useful to use that time next week.
Yeah. Okay.
Any final comments from? No? Thank you again, Jacob and Gary, for being willing to speak to this week, and we'll look forward to seeing you next week. Please help us spread the word about the working group and recruit members. Thank you again.
Thank you guys for all the work you're doing.
Bye bye. Thank you.